Thank you very much, Mr. Chairman. I was telling my colleagues it's going to be the end of the day and the last session, so maybe there will be three people, the three speakers. I'm happy that you stayed on for this particular session. So I'm going to give a summary of what we have been doing regarding harmful algal bloom events in the country. But actually, the activities that we have been doing are related to the activities in the region as well as in the world. Okay, so why study or do something about harmful algal blooms? Recently, meaning the last few decades, the last 3 decades, harmful algal blooms have been causing not only toxicities, like, for example, paralytic shellfish poisoning, the diuretic shellfish poisoning, and amnesic shellfish poisoning primarily. And fish kills is the other type of harmful algal blooms that had been a problem not only in the country but also in the region. You might have heard of what happened to Chile. Lately, they had a massive fish kill that damaged their sardines and salmon industry and cost them billions of dollars as a negative impact. And they say that these HABs, in short, are related to some global and regional environmental changes. And in fact, some people would like to relate this to climate change, including the intensified ENSO in this particular region. In the Philippines and other countries, many have sustained their HAB program, meaning research and development as well as management programs. In the Philippines, it's the Department of Science and Technology that is funding us, primarily the Marine Science Institute, in this particular HAB program, but it is the Bureau of Fisheries and Aquatic Resources that do the management of the problem. Well, of course, the problems related to HABs are linked to food safety and food security and primarily. But of course, they say that there could be environmental impacts like food chains and other impacts to the environment may be considered. So here I have a picture of a Prorocentrum bloom in Bulinaw where we have Center for Marine Studies, and this was the first big fish kill in the area. Prorocentrum has been blooming not only in Southeast Asia, but this is a common bloom in North Pacific as well in Korea, China, and Japan. Now, also there has been a Noctiluca bloom in Manila Bay from 2009 to 2012, and this has been recurring in the recent years, and they say this is due to, well, they say again, to ENSO or El Nino. So this is just a summary of what have been occurring in the Philippines in terms of harmful algal blooms. The first record of the bloom was in early 1900s. They were not sure of the organism that bloomed in that period, but the first pyrodinium bloom, which caused toxicity to several people, was in 1983. It happened in Samar Leyte, and from then on, this particular organism has been blooming in many areas, including the southern part. So Manila Bay is in the northern side, Samar is in the middle part of the country, and even in the southern part, there have been reports of this particular pyrodinium organism blooming, and it causes paralytic shellfish poisoning. So towards this period, meaning lately, we have already recognized some other organisms that can cause paralytic shellfish poisoning and other types of poisoning that I had mentioned a while ago. Okay, I'd like to show you what happened in 2005, and this would show that indeed some harmful algal blooms may be transported by currents. And this happened in February 2005. The mayor of Palawan called us to help in their particular problem of harmful algal bloom. You can see here is Palawan, the beautiful island of Palawan. And the bloom happened on the western side, and it happened from the southern part of this island, southern part of Palawan, and it went up from February to about April, following the current system of the South China Sea during that period. Since that was happening in Palawan, I asked my students what was happening in the rest of the region. My students were in Malaysia, and Sabah particularly, and there was also that bloom of Coclodidium earlier in January and December. So I requested my colleague, Laura, to help me in this regard. And so she came up with this MODIS satellite images, and it would show that the algal bloom came from Malaysia, from December to January, going up to our area, Palawan, in March, and in February, March, and it ended in April, when there was already a change in the current system in that area. I'm sorry, I have a cold, that's why. Okay, so by the end of the period that was in May, there was no detectable bloom of the organism in Palawan. By the way, we were ground truthing during that time from February to about April to really investigate on the organisms that were blooming. And they were the same organisms that bloomed in Malaysia earlier. And what has been shown, this was carried by the current system from Malaysia to the Philippines. Okay, so what has been done in terms of researches in the Philippines? There was almost no work on harmful algal blooms during the 80s and early 90s. The period of real in-depth studies on pyrodinium blooms and other harmful algal blooms started in 93 to 95, and that was the time also when we decided to have the ASEAN Red Tide Network, because there were common harmful algal blooms in the region, and I coordinated that particular Red Tide Network. It was an alert network, an information network, so that the region could benefit from the knowledge from other countries. And now also we were involved in the IAEA study on the application of nuclear techniques to address red tides or harmful algal bloom concerns. The project started in the Philippines. It became a regional project and then a global project. And then it was also the project that really started the development of the RBA to be tested for particular harmful algal blooms. I'm sorry. Can I go back? Sorry. Then in 2009, we came up with a big program. We call it the Philippine HABs. Actually, it started even before that. And it was called the Ecology and Oceanography of Harmful Algal Blooms in the Philippines. This is part of the global program, the Global Ecology of Harmful Algal Blooms. And we're coming up with a book, another book on this. And I have chapter on this, which I co-author with Don Anderson and the rest. It's on life cycles and hubs. So we have understood the biology and ecology of the organism in at least 3 bays, more or less the biology of the organism in 3 bays after doing some in vitro and in vivo experiments. So we're now moving on towards some applied works while we are still doing the basic work together with some oceanographers, like for example, Dr. Villanoy, Dr. Sringan. So it's a team of people doing the work on the molecular biology of the organisms, the taxonomy and ecophysiology of the causative organisms, the toxinology of the causative organisms. We do chemical oceanography, we do physical oceanography, some modeling, like for example, Dr. Villanoy, myself, and some students did the first modeling of pyridinium bloom in Manila Bay. In fact, it was the first modeling of a tropical algal bloom. And then geological, Dr. Sringan is working with us. And then Laura and Alette, together with Cesar Villanoy, are doing some, aside from the modeling, would do some remote sensing and ocean color studies to really understand and help in the management of these blooms. In terms of the R&D for management, the UP-MSI HAB program has been collaborating with some other institutions, like for example, the National Institute of Geological Sciences in our early studies, the Bureau of Fisheries and Aquatic Resources, and the National Fisheries Research and Development Institution, the Philippine Nuclear Research Institute, where we had this study on the use of RBA, the nuclear techniques for detecting the STX, which is now accepted by AOAC. So we started also with that work and help in the development of that particular monitoring tool and management tool. We also coordinate with the SUCs. So therefore, the UP-MSI HAB program is not only a research and development program, it is some sort of a public service or an extension service to the country. So in terms of outputs, we have started the HAB genomics, and as what Professor Concepcion, sorry, why is it? It's moving. Okay. As what Professor Concepcion has said, actually this has been extended because not all of this have been completed in 2015 because of some problems in the development of the molecular and genetic monitoring tools, but some of them have been tested already. So some of the tools have been tested, some are being enhanced, and Professor Luis Ma is part of this team. Professor National Scientist Luli Cruz is also part of the team, and many other biochemists in my institute and also in the other institutions that I have mentioned. So some other chemists are also doing this, the genome sequences, the toxin biosynthesis, and then the HAB detection kits. Now, it is very important that we maintain the cultures of the organisms so that we can do this, and so therefore we have a culture for the tropical species causing harmful algal blooms. And one of these are several strains of pyridinium. Okay, so this is the pyridinium bahamense that I was talking about, and one of the early detection tools that were developed were using protein profiling and they have this further analysis and further enhancement of the tool, and this was done by Professor Luisma and his students. So, by the way, I'd like to mention that we have completed the life cycle of the organisms in vitro, and we considered the life cycle in vivo, and this was helping, these data sets helped us in the modeling of the blooms of the organism. Okay, so we also tried collecting and isolating and identifying the bacteria associated with these harmful algal blooms, meaning the endosymbionts and the exosymbionts. And one of my students who's doing his PhD abroad in Canada, who's coming back soon, has found some very important knowledge regarding the endosymbionts and their role in the life cycle of the organism. So the algalitic bacteria that were taken, isolated from from Pyrodinium, for example, were tested in some other harmful algal bloom species, and indeed some of them were algalitic. And this is also the work of Professor Luisma, and he's using this time genetic molecular tools. Actually, he has already a small gadget for this, and he's trying to test this in the field, and this has been developed for Pyrodinium bahamense species, strains in different areas, like for example, those in Honda Bay, in Sorsogon, and in Samar, and also in Zambales. So the other HABTech program is to have an operational predictive system. This is being developed by the group of Alet. I was thinking she would be coming with us, so I did not include any slide on this. I thought she would be coming, but anyway, Dr. Villanoy here can help us with the models that they have started. They have a descriptive model and predictive model for those that have been studied more in depth, for example, in Sorsogon, and they have been transferred to the Bureau of Fisheries and Aquatic Resources for trial, meaning for prediction. And then we are going to set up also and roll out a clay technology. By the way, we learned this from Korea. We came here and we saw the clay technology for HAB mitigation in Korea, and that was for Coclodinium. But what we studied was the one for Pyrodinium. So what we did was we had several experiments in the lab and did some experiments in the field. The Department of Science and Technology wanted so many statistics, so many data, so many how do you call this, parameter, whatever, I cannot say it very well now. Parameterize the model and so on. So we did many experiments, and he even wanted the amount of clay that is needed per area and so on and so forth. So we did that for specific areas because we know that different areas would have different physicochemical parameters, and the changes would vary from season to season. So all of these were included in the enhancement of the technology. Okay, so this is the first model for the clay technique, but this has been enhanced later. And this one is the spot. By the way, the spot is the one being used for testing the toxin in situ, because this one would be deployed in the field, and the spot, it would be the one that would have the toxin adhere to the resin, and this one would be tested for toxicity later. Okay, so this was one of the experiments done by the group of Dr. McLone on the clay technology, and all the parameters were tested, and it was done away from the coral reef area, because we don't want to damage the coral reef area with the clay. And we had the monitoring for 24 hours every time, and we did the monitoring of all the organisms that we could monitor in the field, what happened to them during the 24-hour experiment. Okay, so this was one of the models done by Dr. Alet Iñiguez. It is the operational predictive system for harmful algal blooms, but actually this is specific for pyrodinium blooms, because this is the main problem in the Philippines. It has affected more than 31 bays in the country. And so here, this is almost 3D model, and what is incorporated here, aside from the physico-chemical parameters, are the life cycle characteristics. So all the data that we have gathered from the lab and the field, so the life cycle characteristics, so from the cyst or the seed stage up to the time that they will form bloom, and up to the time that they will seed again the sediments. Okay, so this is the aim, the dream, hopefully to be done in the next 5 years or so. So hopefully we could have more data. Our dream is to have real-time data. So maybe we can have some help from KIOST in this regard. And then it would enter the model, the heuristic and biophysical model. And then there is also near real-time, how they call this, ground truthing in that sense, because this is in collaboration with some people at the LG, local government units, and the Bureau of Fisheries and Aquatic Resources. So this data would be basically chlorophyll A, temperature, salinity, light and turbidity, dissolved oxygen, and for the toxicity, we will be using the spot. Okay, so the field activities that should be done regularly will be done by the government agencies. We have already trained them in, I think, 3 areas, and we need to work with them again in this new phase of the project to be able to see how they have managed the initial predictive system that had been done, so that enhancement could be put into place. So the goal of the harmful algal bloom program in the Philippines is there are actually 3 goals, main goals: to promote sustainable mariculture, to help protect public health, and to promote seafood export. And this is in relation to seafood safety, especially for in relation to paralytic shellfish poisoning, ciguatera fish poisoning, amnesic shellfish poisoning, and tetrodotoxin poisoning. So here, in the mariculture, site selection would be important. Sediment profile studies, land-based inputs would be important knowledge. There should be monitoring of the culture sites or harvest sites. And here the tools would be very important. And the bloom dynamics of the organism in specific sites should be considered, because we know that blooms are species-specific and area-specific. They have unique characteristics of the blooms per area. You cannot have one size fits all for the management of harmful algal blooms. There could be some generalities, but there are some specifics, especially in terms of the physical-chemical characteristics of the area. And then for the management response, we want to help in fish kills and, of course, the toxicity prevented. And this could be done with the help of some models, descriptive as well as predictive. And so data sets are needed from ground truthing and also from satellite images and many more from previous models. And then mitigation studies are now being undertaken. Okay, so we have actually done some research publications. There are more of this. Some of the ISI publications are well cited, and these are the beginnings of the publication for the IAEA studies. This was done with the PNRI. And this one was the first modeling of a toxic dinoflagellate in the tropical setting. And this one is a well-cited paper. In fact, it became the foremost downloaded article in the Blackwell Synergy for 2004 to 2005. So my co-authors are, one of my co-authors are here, Dr. Seringan. So we have made paper presentations, some as invited in keynote plenary presentations. We have many graduate students. Some of them are going out for their PhD. And we have enhanced our research capability. Like, for example, we have this HPLC, I think 2 of them, confocal laser scanning microscope facility for the causative organisms and many more. And we hope to get some more. So for the regional and international cooperation, we have the Westpac IOC. In fact, it's not only Japan and the other countries that are collaborating with us from the northern side. Of course, Korea has been very active. Aside from this Westpac activity, we have the Northeast HAB network. I have been invited, the only one from Southeast Asia. And then we have the HAB-C network. I also chaired this for a while, maybe 10 years or so. And then we have the Global Ecology and Oceanography of Harmful Algal Blooms of the UNESCO. We have a new one. It's the Global Ecology of UNESCO. So my time is up. Okay, thank you very much. Thank you very much.